Hello again, everyone. This is Dr. West, and this is lecture 7. So again, I'm going to primarily be showing you a pre-recorded lecture by Dr. Bennett, but I wanted to sort of set it up a little bit differently than the way she sets it up. And maybe this is just because this is how my brain works, but I like to sort of see the overall picture first. So let's take a step back and look at the overall picture. So what we are talking about with the nervous system is how it communicates. And even one more step back from there. The reason we even have a nervous system to begin with is to detect what is happening in our environment, and that can be the external environment around us. So that includes stuff like sights and sounds and smells or touch, stuff that lands on us, for example, or touches our skin. Or that can be our internal environment, so stuff that's happening within our body. So for example, if you have a stomach ache, right, you're going to get information about that, like, that doesn't feel good. My stomach doesn't feel good. So the job of the nervous system is to detect what's happening in the environment, send that information to the brain. The brain then processes all that information and then decides if you need to, first of all, respond to that information, if you need to make some sort of movement, or if you need to do something about that information, or if you just need to simply remember that information, or both. All right? So that's sort of the overall goal. That's why we even have a nervous system to begin with. Now, stepping in a layer, right, it does that, right, by converting all of that energy that's happening in the environment. So the light that Dr. Bennett has been talking about that goes into your eyes, the moving air that goes into your ears, and all that kind of stuff, it converts it into the language that neurons and that our brain can understand, which is action potentials. And action potentials, right, another way of thinking about them is that they are a form of electrochemical communication. And I purposely use that word with my students because I like it. It has two parts, right? Electrochemical communication, which means it has an electrical part to the communication and it has a chemical part to the communication. So what we've been talking about in the previous two lectures was the electrical part. The electrical part is the action potential. And your cells, your neurons, can generate that electrical part because of the movement of all of those ions, all that sodium and potassium and all those channels that are opening and closing, right? And then that pump that's having to reset everything. That's how that electrical part is getting generated, right? But we've also been telling you, we've been saying this word synapse a couple times over and over and over again, right? We've been saying that neurons don't actually touch each other. Right? And they don't actually touch, for example, if a neuron is going to stimulate a muscle, it doesn't actually touch the muscle. The end of the axon doesn't actually touch the muscle. There's a little space called a synapse that's there. Right? And if you think about it, we don't want to be shooting electrical energy from one place to another, because if you think about what that looks like, think about clouds in the middle of a thunderstorm that are shooting electrical energy from one cloud to another or from the cloud to the ground, right? It looks like lightning, and we don't want to be shooting lightning around our brain. That's a terrible, terrible idea, right? So we use the second part of our communication system, the electrochemical communication system. So we use chemicals at the synapse, and those chemicals are called neurotransmitters. So this particular lecture is going to start to introduce you to that second part of this particular form of communication that our neurons can do, and we're going to start to talk about the chemicals. So again, before I jump into Dr. Bennett's lecture, I'm going to briefly show you a little bit of what I have recorded here, talking a little bit about the synapse. I'm going to show you another video about the synapse. I will then go into Dr. Bennett's lecture, and then I'm going to interrupt her, and I'm going to show you a really cool video that I found. It's sort of a recreation of an experiment, right, of how scientists learned about the chemical part of neurotransmitters and of neurocommunication, because we knew about the electrical part for a while, but it took us a while to figure out the chemical part, and I'll show you that sort of recreated. So again, first watch this part, and then we will go into Dr. Bennett's lecture. You'll see me add a quick video, and then we'll finish out with her lecture. So happy studying. So now we've come all the way to the end of the axon, and here is the end of the axon. This is just one little part of the end of an axon, because let me go back, let me find a good picture. Let me go back up here to my original picture. See how axons can actually have multiple endings here? So I'm just going to show you one little branch of this axon for this synapse. All right? This particular axon, this particular branch of this axon doesn't actually touch the dendrite of the next cell. There's a gap in between, and that gap is called the synapse. Sometimes it's called a synaptic cleft, same exact thing, right? But they don't touch each other. And we don't want to send that electrical energy. We don't want to be sending, all right, that exchange of sodium and potassium ions, because essentially that's going to create a situation kind of like lightning, and that's bad. All right, so what our brain has evolved to do instead is to send more stable chemicals, which we call neurotransmitters. And we'll get to those in just a second. But I want to cover a little bit more terminology that I've been throwing at you. At the synapse, again, the space is called the synapse. This end, which is the axon end, is called the presynaptic neuron. This is the neuron that is sending the message, that is delivering the message. Right? The postsynaptic neuron, which is usually a dendrite, is the one that is going to be receiving the message. Okay? And the message that it can receive can be one of two messages. It can be excitatory, meaning it's going to depolarize the cell and make it more likely to produce an action potential, right? And an excitatory postsynaptic potential, which is why it's called that, because it's the postsynaptic neuron that's getting, right, the message, right? That's why I waited until now to show you all that crazy terminology. Remember, if you go back up here, right, where I was showing you EPSP and IPSP, that's what I meant back up here, right? But excitatory postsynaptic potential means when it catches a particular neurotransmitter, that particular neurotransmitter is going to cause this neuron, this dendrite of this neuron, to become a little more depolarized, meaning it's a little closer to zero, a little more likely to produce an action potential that will send down this neuron's axon. But some neurotransmitters, when they are released and they bind to the postsynaptic side, are inhibitory, and that's what IPSP stands for, right? This is actually a good thing, though. You don't always want it to send on a message that says go, go, go. Sometimes you want it to kind of serve as a brake or a balance, right, to the excitation. So your body does have both. It has a way to say go and a way to say stop. And so you sort of have this ability to balance itself out. The most obvious example of this that we'll be talking a lot about as we get to it here is someone that has used alcohol. Alcohol that is taken into your body and ultimately ends up interacting with your nervous system acts like GABA. Your body kind of sees it as artificial GABA, and GABA is one of the neurotransmitters that we will be talking about. But when GABA binds, so when GABA is released and it binds to the postsynaptic neuron, it actually inhibits that neuron. It makes it more polarized. It hyperpolarizes it, means it makes it less negative. So it makes it harder for this neuron to then send a message on. And you might be saying, okay, well, I don't get it, Dr. West. Well, think about some of the side effects of alcohol, right? Some of the side effects, again, of just a little bit of alcohol, not a ton of alcohol. Right, so not somebody that's passed out, although technically that's the same thing. But think about somebody that's had just enough, they start slurring their speech. Start stumbling around a little bit, right? That's because all the neurons are not firing correctly to make those fluid motions. All the muscles aren't getting all the same messages at the exact same time in the exact same way, right? Because there's a bunch of artificial GABA in there sending improper inhibitory messages to some of the neurons, so it's not getting to some of the muscles in quite the exact same way, and the person is exhibiting some of these weird side effects. Right, so you need this balance, right? But again, alcohol can actually act as too much inhibitory, right? But that's just an example. We'll talk more about that later. But hopefully these terms make sense.
Communication between two neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
Welcome to Lecture 7. We're going to start Chapter 5 here, and again, this chapter is also broken up into two parts, just like Chapter 4. So that means, hint, these two chapters are pretty important since they're getting two different lectures and time devoted to them. As you're studying for the exam, think about them. And to this point, we've talked about how neurotransmission occurs within a neuron, and we're going to now move that next step, like we ended the last lecture, talking about the neuromuscular junction, right? So that was the nervous system talking to a muscle. Well, now we're going to talk about how does a neuron go from communicating within itself and the action potential to the neuron next to it. So we're kind of coming back. As I mentioned earlier on, if you recall, that it's difficult to teach neurotransmission because it's cyclical, and you just have to pick a part to step into and start teaching it, and then try to circle back. So now we're going to talk about how, once that action potential reaches the end, where does that go? Where do we go from there? What happens? One, you have to think about there are important pieces that have to be present, and we're going to talk about through some of those things. So how does neurocommunication actually occur between neurons? One of the things that has to be present is there has to be a receptor to receive information on the postsynaptic cell, and it has to be sensitive to the same chemicals that the presynaptic is outputting, right? That's releasing. So if a neuron's releasing a chemical that another neuron near it can't understand, then there's no feedback, there's no connection, there's no support, there's no creation of a connection. So the two neurons have to be talking in the same language, right? Same code, and this chemical will either have one of two messages, right? It's going to be excitatory or inhibitory. I'm going to keep driving that home. That is the only two messages, 0, 1, whatever. If you think about computer code, stop, go. I mean, that is literally the way our bodies function. It's that simple of a code. I mean, it's complex because it's arranged in lots of different ways, but that's basically the ending, or the kind of basis of everything that we do. Our bodies function, or our nervous system functions. So we're going to talk about an example to help give context and helping you piece all these things together. And again, we're going to talk about the heartbeat. So again, this is the nervous system communicating to a muscle. So we're talking about things that happen outside, but this is where we kind of found out about chemical messages, and it will help you understand. Hopefully, it will help you understand. So what we can see is that in the body, in the way that our bodies respond to the world, we see and we can actually feel times where our heart rate speeds up, and we have times where our heart rate's slower. And we're going to talk about what are the different chemical messages that might be driving this type of changes in muscle contraction. So this comes back almost 100 years ago now, where we were, Lotto Lowy was playing with a frog heart and was examining the role of actually the vagus nerve. If you recall back, vagus nerve is one of the 10 cranial nerves, one of the 12 cranial nerves, it's number 10. And it controls a lot of the peripheral organs as a major role in the parasympathetic nervous system. So he has this frog heart, and he's checking out and playing with the vagus nerve to see how what its role is on the heart, basically. The vagus nerve releases acetylcholine as its neurotransmitter. He didn't know that, but we know that now. And what the work he was doing we're going to kind of go through it step by step. 2 hearts. They're in separate containers that are connected by this fluid tube, this tube that allows fluid to transfer. 1 heart has a vagus nerve attached to it. He's got a stimulator on it. It's not been stimulated yet. We're going to talk about what happened. Then he's got another machine over here that is detecting the contraction rate of the muscle fibers of the heart. So you see the heart is beating at a certain rapid rate. Heart 2 is also beating at that same rate. The heart will contract actually rather rapidly when the vagus nerve or the parasympathetic is inactivated. He goes and he stimulates the vagus nerve, and as you'd expect, the heart slowed down. Heart 1, we're directly activating the vagus nerve that's attached to it, and it's causing changes in the contraction rate. What do you think is happening over here in heart 2? Do you think it decreases? Do you think it remains the same? Well, if you guessed that it decreased, you're right. This is how we discovered that the vagus nerve is releasing a chemical to cause these changes. And this chemical, following a concentration gradient, will disperse, and it will have the same impact on another heart. Kind of cool.
All right, so where we're headed is this. Let's start talking about the chemicals that we send across this space called the synapse. We didn't always know there were chemicals. We always sort of knew about the electrical part, right? Because for a long time we knew at least about some other peripheral organs, particularly things like hearts. We knew that you could electrically stimulate them, and we knew that there were nerves that electrically stimulated them and made the heart beat. So we sort of knew about that, but we weren't really sure about the chemical part. And it wasn't until a pretty cool experiment done by the German physiologist, Dr. Lowy. He was the first that actually convincingly demonstrated that communication across the synapse actually is by chemical means. Now, the video that I'm going to show you sort of makes it sound like it wasn't really his idea, but no, it was totally this dude's idea, and he got all the credit, and he won a Nobel Prize for it. So I don't want to try to downplay his participation, but I think this video is pretty neat that I'm going to show you, because it sort of demonstrates how he was able to figure out that it was a chemical that was actually involved in the process of neurotransmission. And again, ultimately those chemicals went on to be called neurotransmitters. He called them vagus stuff because he was stimulating the vagus nerve that led to the heart. But ultimately we went on to call them neurotransmitters. Again, neurotransmitters in general are just chemicals that travel across the synapse and allow for communications between neurons. But very quickly, watch this video about his experiment and see what you think.
At the end of the 19th century, one of the great puzzles challenging scientists was how the billions of nerve cells that make up the human nervous system communicate with each other. They knew that each nerve cell, called a neuron, carries electrical impulses, and many believed that the connection that allows the impulses to jump from one neuron to another was electrical as well, until our next great discovery. In the early 1900s, British biologist Henry Dale was conducting a series of lab experiments studying the physiology of nerve impulses. In one of his experiments, he injected adrenaline into a cat, expecting the animal's heart rate to increase, but nothing happened. Only Dale realized his mistake. It was the same cat that he'd already given another drug to, one that slowed the heart rate. With this mix-up, Dale realized he was onto something. If a drug could interrupt the nerve impulses that govern the beating of the heart, then the connection between neurons must be chemical, not electrical. Dale turned to his colleague, German biologist Otto Lowy, for help. To test Dale's hypothesis, Lowy conducted an experiment using 2 frogs. Lowy put the first frog in a saline solution. Then he electrically stimulated its vagus nerve, the nerve that controlled its heart rate. The frog's heartbeat slowed. Next, Lowy took some of the solution and applied it to the heart of the second frog. What happened next was extraordinary. Without any electrical stimulation of its vagus nerve, the second heart slowed as well. This was the moment of discovery. Lowy realized that the vagus nerve of the first frog had released a chemical which had directed the frog's heart muscles to slow their contractions. Here was proof that the transmission of impulses among nerve cells and then to the heart muscle was chemical. Lowy had discovered the existence of the first known nerve chemical, now called a neurotransmitter. Today we know there are many neurotransmitters in the human body, and researchers are using that knowledge to learn more about how the brain works and the chemical messages it sends. For example, low levels of serotonin have been connected with depression, alcoholism, and anxiety disorders.
So acetylcholine is responsible for activating muscles. We talked about that at the neuromuscular junction, that when it's released, causes the muscle to contract. But we just talked about how acetylcholine causes the heart to slow down, which means it's stopping or inhibiting contraction. One of the things that's really important to understand, and we're going to talk more about neurotransmitters in the next lecture, neurotransmitters do not have there are only two. There are only two neurotransmitters that have a blanket effect on the body. All the other ones, it depends on the type of receptor it's binding to, and it also depends on where it's binding, like what part of the body it's binding in, about how it's going to actually have an impact, or what type of impact it's going to have. So on heart muscle, acetylcholine causes it to relax, not contract. And on your skeletal muscles, it causes contraction. Same messenger, but because of the type of receptor it's binding to, and then the natural effects of that receptor being bound, causes an impact. All right, so other things that we learned from his research includes epinephrine or adrenaline, depending on what part of the pond you're from, what side of the pond you're from, you will call it one or the other. In the United States, we call it epinephrine. In Europe, they call it adrenaline. This is that chemical messenger supports fight or flight. We will spend a lot of time talking about that when we talk about stress in part 2. There's also norepinephrine, which is also known as noradrenaline. It is a neurotransmitter that's primarily found in the brain, but it also plays a role in the autonomic nervous system, and we'll talk about it. All right, so what is a neurotransmitter? Neurotransmitters are chemicals that are released from a neuron, and its target is close, and it's typically another neuron or a tissue. And it will have either an excitatory or an inhibitory effect. Once the neurotransmitter is outside of the central nervous system and the peripheral nervous system, and it moves into the bloodstream, it becomes a hormone. And we'll talk about hormones in the next section. But I just want you to understand that neurotransmitters work in a very small, they have very close targets, and they have typically a very short-lived effect. Hormones are the opposite. They have very distant targets, and their action may take a lot longer for them to have an impact. So hormones are kind of the antithesis of neurotransmitters when we're talking about functionality and chemical messaging. Right, moving into the synapse, which is really important. There are a bunch of definitions listed. I'm not going to go through each one of them, but recognize that there are key players. You have to have a pre- and postsynaptic neuron for communication to go across a synapse, which is the opening between them. There are synaptic vesicles, which can be packaged together and be put into a storage granule. And the last thing that's really important you must have is that the postsynaptic neuron has to have receptors for the chemical message that's being released by the presynaptic so that they can communicate, right? So same language. If it's sending a different message and the postsynaptic can't interpret it, there's no point in having that communication occur. So here's a picture, just so you can see in real life or electron microscopy. This brown here is the cell membrane. It's labeling where there is some phospholipid bilayer. You'll notice all the vesicles are made of the same material. The inside contains the neurotransmitter, but the outside is the same thing as the cell membrane, which is why when the neurotransmitter is being released, the vesicle actually combines with the cell membrane. And this is why, because it's made of the same material. Now I know you can't really see it, but trust me, there is a small itty bitty cleft or spacing that goes on. So this is the pre, talking to the post. This is the axon. This is the dendrite. Here's a caricature of what's happening. You have your presynaptic, which is releasing its neurotransmitter. The neurotransmitter diffuses across, and due to the concentration gradient, moves towards the dendritic spine and binds to the receptors that are present, and then causes a change over here. We're going to talk about all these steps. All right. There are 4 major steps. The First is that the neurotransmitter has to be synthesized and located in the axon terminal. Then it needs to be transported to the synaptic membrane to be released into the synaptic cleft, or exocytose, that's another word. And then goes across and it binds and activates the target receptors on the other neuron. And that will continue forever unless the neurotransmitter is inactivated or removed in some way. We'll talk about all these steps and how the body works with them. So the synthesis and storage. Typically, neurotransmitters are synthesized in either the axon terminal or in the cell body. And it depends on the type of neurotransmitter, and when we get to neurotransmitters and talk a little bit more about them, we'll talk about where they are actually created and how they get sent to where they need to be. But here you can see that the axon terminal, if it's created there, it hangs out there, it stays there, it gets packaged, it gets sent out, it gets repackaged, sent back out. And then in the cell body, if that is involved, it's involved because the neurotransmitter is coming from the DNA, and then that's actually the proteins are being created by the genetic code, which then it is packaging them into and communicating them through ribosomes, and then the proteins being created and being packaged through the Golgi apparatus, and then it's being sent down the microtubules. Presynaptic, these are vesicles filled with neurotransmitters that's coming from the cell body. They're being packaged. They're being told to be released. They're being released. The neurotransmission is, neurotransmitters are then binding and unbinding, binding and unbinding, activating the receptor, and then inactivation occurs, and there's a couple different ways. We'll talk about all of these momentarily. Step 2 is how do we get the neurotransmitter to be released? Well, this happens at the axon terminus. The action potential causes voltage-gated calcium channels to open. Calcium's now playing a role. So the action potential comes flying down the axon, and when that positive 30 hits the axon terminus, the calcium channels, which are sensitive to the positive charge, open. This causes a rush of calcium into the cell, and then calcium actually goes on and binds to a few other chemicals. I don't care that you learn them all for this class, but when it makes this complex, it then binds to the vesicles, and it starts to basically propel them, move them towards the cell membrane so that the neurotransmitters that are present inside them can be exocytosed or released into the synapse. If they're bound to those microfilaments because they came from the cell body, then it will have them come down and replace and empty their contents as well. So here's an example. Action potential comes flying down, calcium rushes in, it either pulls the vesicle from here or the vesicle's just floating around in here. The calcium ions bind with calmodulin, which makes a complex, which then causes the vesicles to migrate towards the cell membrane, and then the neurotransmitter is being released. For that neurotransmitter to have any impact, there has to be a receptor on the postsynaptic cell that is sensitive to the neurotransmitter that's being released. And there are 2 types of messages that can be sent. There are depolarization, which is the EPSP we talked about, or hyperpolarization, which is the IPSP. Only 2 messages that can be sent. So when a receptor is activated, it's going to receive one of those 2 messages. Now we're talking about the postsynaptic receptors. There are also presynaptic receptors, and those are primarily used for negative feedback. Not all receptor systems, not all neurotransmitter systems use them, but we will talk, and I'll highlight the ones that do. So there are receptors both on the postsynaptic neuron and the presynaptic neuron. Here's a picture. Neurotransmitter is being released. It's going out here, and it's binding to the receptors. These receptors are changing their shape, becoming more permeable to either sodium or chloride ions. Either one, it doesn't matter, depending on what the message is. It's important to note that the neurotransmitter does not get absorbed by the postsynaptic cell. Let me say that again. The neurotransmitter is not absorbed or brought into the postsynaptic cell. It only binds to the receptor and then is released. So inactivation has to occur through different processes, and we'll talk about that in a moment. There are presynaptic receptors over here, and they're usually on the outside or away from the site of action, so that as neurotransmitters, it's binding and releasing and binding and releasing, and this neuron is releasing more and more neurotransmitter. At some point, there's going to be enough that it starts to diffuse outside, and when it does that, it can bind to that receptor, and then this cell receives a message. Halt, there's enough neurotransmitter, you can stop sending your message. Your message has been received, and it shuts off the presynaptic. That's that negative feedback piece. So now we're going to talk about the inactivation occurs at least 4 ways that we're aware of at this point. There are 2 ways that are considered passive, and there are 2 that are considered active. So the 2 passive ways of removing the neurotransmitter is by diffusion out of the synapse, following concentration gradient, it just moves away from itself, or it can be taken up by surrounding glial cells, support cells, typically it's astrocytes, absorb neurotransmitter out of the synapse to get it out. And the 2 active ways are degradation by enzymes and reuptake. So that's literally pulling the whole neurotransmitter back up into the presynaptic neuron. That's what reuptake is. So again, those are your 4 ways. 2 passive, 1 and 2 are passive, and 3 and 4 are active. Active because it's involving the synapse, or the 2 neurons are involved in the synapse, right? 3 fantastic videos for you guys to watch. They all share important information that highlight different areas and bring information into different pieces and talk about them in different ways. So enjoy those, go watch them, and then when we come back, we'll talk about the different types of synapses. All right, so welcome back. Hopefully you enjoyed those videos. If you didn't watch them, make sure you do watch them. Again, those links are available on the help page. So the nervous system has a variety of synapses. We're going to talk about them briefly. Part of it is to help us understand how so many different types of messages can be sent within the nervous system. The 2 types that we're really going to focus in on are looking and talking about axons talking to dendrites, and then the 2 different types of messages they can send, which are excitatory and inhibitory. Those are all I really, really want you to focus on and gain greater understanding. But if you're really excited about this material, I'm going to give you some more additional information. So I'll try to highlight when things are critical for class and when things are more critical for your interest in this material. So outlined here, you can see that there are multiple ways in which the nervous system and neuron can interact with the world. There are dendrite to dendrite, there's axon to dendrite, which is what we are communicating or what we're learning about. So this one's really, really important. This is where most of it happens. There is axon to extracellular, and this is often, at times, neuromodulators, things that are not trying to drive necessarily specific communication with the neuron. There's also axon to the cell body. There's axon onto axon. It's amazing, all the different places. And there's also axon and synapse, and then axon, 2 axons in the same synapse, and then there's axon onto axon. And then the last one here is one that we'll talk about when we get to movement and we talk about hormone release, but this is where the neuron will directly release its contents into the bloodstream or into a muscle or some other type of tissue. But that's an important role that it plays. Now we've talked about physical synapses, and those are ones where, or chemical synapses is another way to talk about it. That's where the neuron, the postsynaptic, changes the information into a chemical, which is a neurotransmitter, which then is released and put into the synaptic cleft, which then binds to receptors on the postsynaptic and changes the permeability of that postsynaptic, right? So that's that process. That is a chemical synapse. It's not highly efficient when you're talking about the neurotransmission that occurs via the action potential or via the exchange of ions or the electrical polarity changes. That is very, very, very efficient. This physical change, it's still milliseconds. We're still talking milliseconds, but it is slower. It's like 10 to 20 milliseconds versus 1 to 2. Right? So the action potential is very quick. There are a few places in our body where neurons are actually fused together to allow for a concerted integrated communication, and those are created via gap junctions, which is where there are proteins that cross both cell membranes and allow the positive sodium ions to flow directly from one neuron to the other. Very rare. They do exist. They're very fast, but they're not very flexible. They can really only send one message. So chemical messages using a neurotransmitter are slower, but they are also more flexible, in that they can either amplify or diminish a signal because you might have other impacts, other influences around you. These electrical synapses are just immediate, they're fast, and they only have one message to send. It's either go or stop. All right, so there are 2 types. As I mentioned, there are 2 types of synapses. There's the excitatory and there's the inhibitory. Type 1 is considered the excitatory, and this is typically associated with dendrites, and the vesicles are round, so we're talking about the actual shape of the vesicles. The material is very dense. It's a wide cleft, meaning that There's a lot of potential connections that can occur, allowing for a large active zone. Type 2 are the inhibitory, and they look almost the exact opposite, right? They're typically located near the cell body or on the cell body, therefore they're more potent, right? Think about that. Think about placement. The vesicles are flat. There's not as much material being released because of how potent they are. The cleft is narrow, which means they're super close. And then there's a small active zone. So let's look at these pictures right here. So type 1, again, are up here in the dendrites. You have this wider cleft, more action, maybe more synapses can be involved, more axon terminal can be involved. Large active zone, look how many receptors there are here. Lots of places for the neurotransmitter to bind. The receptacle, the vesicles are round, and lots of material being released. Partially because it's a wide cleft, right? If it's a wide cleft, it's more distance to cover, you need more neurotransmitter. Does that make sense? Now type 2, which are the inhibitory, which are also located more likely to be on the cell body, the vesicles are flat, they don't release as much, but the active zone is also smaller. And it's because this is a more potent message. So you don't need as much of this message, hopefully, to shut a cell down, right? So those are the two different ones. So there are a variety of different neurotransmitters. There's upwards of 50 that have been identified. Some can be inhibitory in one location, excitatory in another. I kind of mentioned that earlier. A neurotransmitter there can be more than one neurotransmitter at a single synapse. There's no one-to-one relationship between a neurotransmitter and a single behavior. No matter how much you read about or watch movies and they tell you that this neurotransmitter creates this behavior, or a belief that depression is related to not enough serotonin. Not that simple. Never has been, never will be. So how do we identify a neurotransmitter? A neurotransmitter is something that is synthesized in a neuron. It must be released and have a target close to it. If we introduce the same structured chemical in an experiment, it will have the same impact, and that there is a mechanism for removing it. That's a critical piece to defining the difference between a neurotransmitter and, let's say, a neuromodulator, is that there is a way of removing it from the synapse to shut down its effect. Let's go back to number 3. What we're talking about is a drug, could be an example. So let's use nicotine. It is present in tobacco products. It looks like acetylcholine, and it binds to acetylcholine receptors. Therefore, we know that acetylcholine is a neurotransmitter because of where those receptors are. All right, here's just a picture for you. Here are a couple other factors about what neurotransmitters might do. And on the note, I'm going to leave it. There is another video of me hand drawing the synapse and taking you through each step of what goes on in the synapse. I highly, highly, highly recommend that you review that. It's something I do in class. We draw it, we talk through it, and all those fun things. I suggest heavily that you do that, review that video probably multiple times. All right, thanks.